Impeding ABCB10 protein in liver cells ensures against high glucose, greasy liver. Another multi-establishment study drove by a group of analysts at the David Geffen School of Medicine showed that impeding a protein called ABCB10 in liver cells secures against high glucose and greasy liver infection in hefty mice. Besides, ABCB10 movement incited insulin opposition in human liver cells. The discoveries are quick to show that ABCB10 transports biliverdin out of the mitochondria, the cell's energy-producing forces to be reckoned with. Biliverdin is the antecedent of bilirubin, a substance with cancer prevention agent properties. Therefore, ABCB10 transport movement causes an expansion in bilirubin amalgamation inside liver cells going through greasy liver sickness. Foundation. Non-alcoholic greasy liver illness is firmly connected to stoutness and different problems identified with insulin obstruction and is getting progressively normal all through the world, influencing an expected 100 million individuals in the United States. The liver channels all that individuals devour and sorts it for the supplements that will remain in the body or for the poisons that it will oust. The liver is likewise one of the organs most extravagant in mitochondria, the little organelles and cells that convert food into usable energy through a cycle called digestion. Subsequently, the mitochondria produce significant degrees of free revolutionaries, just as cancer prevention agents to keep these free extremists at sound levels. Both free extremists and cell reinforcements assume a critical part in managing digestion and are raised in insulin opposition and greasy liver illness. One of these cell reinforcements is bilirubin, a yellow bile substance that is delivered from the breakdown of biliverdin, its green bile forerunner. Bilirubin is delivered at undeniable levels in livers from individuals with greasy liver infection. Both biliverdin and bilirubin are discovered normally in the body and delivered during the breakdown of heme, the dark red iron-containing particle in red platelets, which can be found in the changing shade of wounds, from green, biliverdin, to yellow, bilirubin. In vivo insurance from arising SARS-CoV-2 variations by protein nanoparticle antibody. Subduing a protein that works with DNA fixed to potentiate chemotherapy. New non-invasive biomarker can be utilized in the determination of NAFLD. Past research set up that gentle expansions in blood bilirubin substance could be related with security from metabolic sicknesses. Nonetheless, the impacts of bilirubin content inside mitochondria and their relationship to greasy liver illness and insulin obstruction stayed obscure. This flow study shows that expanded bilirubin content inside the mitochondria driven by ABCB10 movement is adding to greasy liver infection. Strategies. In the examination, the specialists eliminated the ABCB10 protein specifically from the livers of mice to test whether ABCB10 expulsion affected the capacity of hefty mice to endure glucose, in the event that they created fat in the liver, and how well the mitochondria in their livers were attempting to change over supplements into usable energy. In lean mice, the analysts discovered no distinction in digestion and well-being when ABCB10 was eliminated from their livers, while in large mice they found that eliminating ABCB10 ensured against insulin opposition and greasy liver illness. Besides, the analysts estimated bilirubin in the mitochondria of liver cells utilizing fluorescent sensors, just as testing cleansed ABCB10 to figure out what ABCB10 transports. They found that ABCB10 transports biliverdin out of the mitochondria and builds bilirubin creation in liver cells, with ABCB10 expulsion diminishing mitochondrial bilirubin substance to levels saw in lean mice. Thirdly, the analysts found that when they re-established bilirubin content in the mitochondria, the advantages on the capacity of mitochondria coming about because of the expulsion of ABCB10 were turned around. Effect. These discoveries shed light on the pertinence of the relationship of some hereditary ABCB10 variations with insulin opposition in type 2 diabetes. While still ahead of schedule to make any inferences, these discoveries could motivate the advancement of treatments that target ABCB10 or mitochondrial bilirubin in the liver to invert greasy liver infection in large people.